Okay, everyone, this is the second half of the video lecture for the chapter 17 on species. If you recall, at the end of the first half of this chapter, the video lecture for it, we were talking about prezygotic barriers. These are barriers that prevent formation of hybrid zygotes. So how do you get hybrid zygotes? You have individuals of two different species or two different gene pools at least that mate. Those are called heterospecific matings, okay? Heterospecific meaning different species. And they can result in a hybrid zygote. But if we have prezygotic barriers kicking in, those are things that prevent formation of the zygote if a heterospecific mating attempt occurs, all right? But now we're going to talk about what are called postzygotic barriers. These are barriers that kick in as sort of a fallback mechanism to prevent gene flow between distinct species or distinct gene pools, even if you do get a hybrid zygote formed. And the way they generally work is that if you have a hybrid zygote formed through a heterospecific mating, the hybrid zygotes will either have reduced survival rates or typically reduced reproductive rates. And so selection operates against them because they're less fit in one way or another. And so these postzygotic barriers then effectively prevent introduction of genes from one species into the gene pool of another. All right, so we have a number of different types of these postzygotic barriers. The first one is called hybrid inviability. Okay, hybrid inviability. So here, hybrids may have high mortality, for example, as very often as embryos. Embryos may not survive due to unequal chromosome number in the sperm and egg, or due to other problems associated with improper development. Unequal chromosome number, where the two parent individuals, because they're members of different species and they have different numbers of chromosomes, very often is a death sentence for an embryo. So that's an example of hybrid inviability. Another example, if hybrids actually do successfully develop into adults, they may be sickly or compromised in some other way, so maybe they're just not viable as individuals from a biological perspective. We also have another one called hybrid sterility. And this is probably the one that most people would tend to think of. So the sterility of hybrids can often be caused by chromosomal differences between the species. It may also be caused by simple differences between the genes, where the genes simply aren't compatible. The gene combinations from two parents of different species very often are incompatible in terms of their interactions. A lot of genes actually within a species work together in what are called co-adapted gene complexes. And genes from different species are probably not going to be compatible in that way. And so when you have hybrids that are formed, in a lot of cases, one of the reasons why they're inviable or sterile is that their co-adapted gene complexes of their parent species have been broken up or disconnected from each other. And so they're simply not viable, or in many cases, they're flat out sterile. All right. Now, let's talk about hybridization in particular. So, on occasion, heterospecific matings can produce an offspring that's effectively viable and capable of producing its own offspring. So, these are the kind of hybrids that we're talking about now for the most part. When you have the geographic range or distribution of two distinct gene pools or two distinct species that come into contact with each other, like, for example, the crows in Europe that we talked about in the previous part of this video lecture, you can get what's called a hybrid zone between them, or where they come into contact. And so this is sort of an area of overlap, you might say, between the gene pools of those two species. And so this hybrid zone then is a region where two genetically distinct populations meet and where the individuals of those two populations potentially mate with one another. So within that zone, you get hybrids produced. All right. Now, in a lot of cases, the hybrid zone may actually result in a cline. So remember, we talked in an earlier chapter about clines. A cline is a relatively abrupt change in the frequency of one or more alleles over some geographic or climatic gradient. Remember that? Okay. So the hybrid zone may result in a cline if a locus or a character state changes over that hybrid zone. The hybrid zone actually might be quite narrow, which can result in a very steep cline if that's the case. So we have two types of hybrid zones. We have, first of all, what's called a primary hybrid zone. This is a hybrid zone that develops within a continuous population as a result of a rapid change in some sort of environmental variable. It could be temperature, altitude, rainfall, I mean, who knows, right? It could be oxygen concentration, pH. So selection will change allele frequencies over these short distances. That's the cline, the rapid change in the allele frequency over that distance. And individuals on either side of the cline can mate and produce hybrids. So the idea here is that we have the cline being coincident with the hybrid zone. They line up with each other sort of geographically, okay? All right. Now, that's when you have a continuous population and the cline results from from an environmental gradient of some sort, and that results in essentially two genetically distinct populations on either side of that cline. Even though they're continuous, they can get together at that cline, which is the hybrid zone. They are separate in the sense that you have certain alleles very abundant, let's say, on one side of the cline and less abundant on the other, and vice versa. That's because selection's operating on them differently as a result of the cline. And what you get in the middle is a hybrid zone because individuals on either side of the cline can get together in the hybrid zone and mate. All right. Now, that's a primary. A secondary hybrid zone is when you have two formally separated populations that expand their ranges until they meet one another. Interbreeding and hybridization can then occur. So we talked in the previous video lecture about parapatric and sympatric relationships. And we said that if you have, for example, one population here, that's its geographic distribution, and a second one here, and they just sort of bump into each other here at their borders, that's referred to as a parapatric situation, okay? We can get a hybrid zone formed there, all right? However, we can also have a situation where you have one geographic range here and another one here, for example, and you have this area of overlap. That's sympatric. So that area of overlap where you have the two ranges actually overlapping one another, coming into contact like that, that's a sympatric relationship. Now, what if you have a continuous geographic range, something like this, and something happens, like maybe a river changes course or mountains are built over long periods of time, and something bisects that range? Well, now you have two separate populations, one on the left, one on the right, that where individuals can't get across that geographic barrier, and now that is referred to as an allopatric situation. Allopatric means other country, or two separated populations, geographically separated with no gene flow between them. Okay, so think about it this way. If this barrier remains long enough, these two populations, which initially were the same species, could evolve along separate or distinct evolutionary trajectories and eventually become different species because their gene pools don't mix. And we talked about the homogenizing effect of gene flow, right? So if that barrier remains long enough, we could have two separate species evolving from the one as a result of that barrier. And then what if that barrier goes away at some point in the future? What if the river changes course again or dries up entirely? And now you have individuals capable of moving back and forth across it. You would get a hybrid zone, and you would get heterospecific matings in that hybrid zone. But this would be a secondary hybrid zone because it's a once continuous population that was split into two. The barrier went away, and during that time when the barrier was there, the two populations evolved separately. The barrier goes away, and they come back together again a second time. That's why it's called a secondary hybrid zone. Okay, so we expect actually to see different patterns of allele frequencies regarding clines, whether we have a primary or a secondary hybrid zone, as we'll see here in a minute. So let's talk about that. In a primary hybrid zone, the expectation is that selection will affect different alleles in distinguishable ways, causing the geographic position of their individual clines to vary. This means that if you look at one allele, you're likely to have a cline located in a certain position, a certain geographic position over the geographic range of those two species. Okay. If you look at a second allele in a primary hybrid zone, that doesn't necessarily have to be, its cline doesn't have to be in exactly the same spot, because selection may operate on it differently in relation to that environmental gradient that sets up the cline. A third allele could have its cline in a different geographic position relative to that gradient. Okay, so the expectation then with primary hybrid zones, again, is that selection will affect different alleles in different ways, and that will cause the geographic position of their clines to vary. They won't all be coincident with each other. They won't all overlap or lie on top of each other in exactly the same spot. All right, we'll see some graphs for this here in a minute. Also, in the case of a primary hybrid zone, any selectively neutral allele, one that selection is not operating on, would presumably not show the cline because selection is not operating on it. That makes sense, all right? Now, what about a secondary hybrid zone? Here, that hybrid zone should reveal a pattern of clines for individual alleles or characters that are actually coincident. They all should sort of line up with each other. Remember, in a secondary hybrid zone, you had that geographic barrier go away, and that barrier was in a geographic location. And now individuals can move into the area where that barrier was from both sides, from both gene pools, start to mate. And so you'll have the hybrid zone located where it is. That's a secondary hybrid zone. But you had alleles moving to different frequencies in one population versus the other on either side of that geographic barrier. So when they come together, they should all have clines located at that hybrid zone where that geographic barrier used to be. All right, they should be located about the same geographic position. Hybrids here will have low fitness resulting from heterozygote disadvantage or disruptive co-adapted gene complexes, that sort of thing. By the way, that disruption of co-adapted gene complexes I mentioned before, that's also referred to as epistatic incompatibility. So epistasis is where two or more genes work together, or one gene controls the expression of another gene, that sort of thing. And in the case of co-adapted gene complexes, you have one or more genes maybe controlling other genes, and that relationship gets broken up in the case of hybrid zones, okay? And that's epistatic incompatibility. All right. So the thing about these secondary hybrid zones where hybrids have low fitness, they're known as tension zones, okay, tension zones, since they help to sort of reinforce the genetic differences between the two populations. Neutral alleles in a secondary hybrid zone are likely to be coincident with other non-neutral alleles simply because their frequencies in the two allopatric populations were likely to have been different in the first place before the populations even joined, okay, as a result of drift and that sort of thing. Think about neutral theory, like we talked about in that chapter, right? So the thing about these hybrid zones is that gene flow will eventually smooth out the cline in the neutral alleles because they're not being selected for or against, so they will eventually bleed across that hybrid zone as the hybrids start to be produced. Because selection is not operating on them, that cline will eventually go away. So let's take a look at what I'm talking about here. So on the left, we have an example of the clines we would expect to see in different alleles, or different genes rather, in a primary hybrid zone, right? So here we have gene A, gene B, and gene C, and you can see that we have the geographic position from west to east along the x-axis, and their clines for the different genes, different loci, are actually located in different positions, okay? And so remember, in a primary hybrid zone, you have one continuous population that's spread out across a geographic gradient, and different genes will be affected by selection differently in response to that gradient. So you wouldn't expect that the clines for those different genes would all be in the same spot along that east-west gradient, okay? And you can see here for a selectively neutral allele, its frequency really isn't going to change. There's no obvious cline in that at all. Now, in the case of a secondary hybrid zone where we had allopatric populations that have been separated for a long period of time, and you had their frequencies or frequencies of alleles or genes in them being very different to begin with, all of a sudden they're thrust back together in that hybrid zone when that barrier goes away. And so when we have locus A, B, C, and D, we can see that for A, B, and C, they all have, all their clines are effectively coincident, right? This is the center of that hybrid zone. That's where that barrier, geographic barrier, used to be. And so the idea is that when that barrier was present, genes A, B, and C went to high frequency on this side because selection favored them there, and other alleles of those genes went to low frequency on the other side. But then all of a sudden they're thrust back together when that barrier goes away, get hybrids between them. Here's the hybrid zone, and now you can see how abruptly their frequencies change, and they all change in the same spot. That cline is coincident across all of them because that's where that barrier used to be. Now, selectively neutral allele might have been higher to begin with on the east side and lower to begin with on the west side, and so you might see a cline in that one, but it's not going to be nearly as sharp as these alleles that are under selection pressure. Okay, so do we see any evidence of this in nature? Yes, actually, we do. There are a lot of cases where this has been documented. Here's one. These are two species of European frogs, and you can see that they do look very similar in terms of their color pattern, the splotchy pattern, I should say, but their color pattern is quite different, all right? They are very closely related, and when researchers looked at 6 different loci for 6 different enzymes or proteins, what they found was a rapid or a fairly steep cline, I should say, right there. This is the center of the cline, and you can see that on, let's say the east side of it. I'm not sure how this is laid out. It could be longitudinally or latitudinally, but anyway, let's say the east side of it, you see that we have the frequency of these alleles being very high. They drop rapidly during the cline and go to very low frequency on, let's say, the west side of it. And if you take a look, go back for a second, that looks a lot like what we see right here, doesn't it? Yeah, and so this is a really strongly documented, well-documented case of a secondary hybrid zone. These two frog populations used to be one continuous population, some sort of geographic barrier, not sure what it was, separated them for a long period of time, during which time they basically evolved into separate species. The barrier goes away, and now we have this cline here between them where hybrids are formed, and you see how that cline's in the same spot for 6 different genes, 6 different loci. Now that's molecular data. What about phenotypic data, so morphological data? Here we basically see the same thing. These are 7 different morphological characters, and we see that cline's in the same position as it is in the case of the molecular data. Okay, so very good evidence of a secondary hybrid zone there. Okay, so why do we have tension then in a secondary hybrid zone? Remember, tension is an effect that basically sort of prevents or at least limits the amount of bleeding over of different alleles across those hybrid zones, sort of acting to keep the two populations on either side genetically distinct. So why is that? Well, this can be a little bit complicated, so I'm going to write out some of this here in a minute. Let's imagine, though, that population 1 is fixed for allele A1, all right? And population 2 is fixed for allele A2. Hybrids that are formed in the hybrid zone between them will then obviously be A1, A2 heterozygotes, right? Okay. Presumably, they'll have low fitness due to heterozygote disadvantage or epistatic incompatibility, like I talked about, breakdown of co-adapted gene complexes, all right? Okay. Now, these heterozygote hybrids can actually backcross with homozygotes of either population to some extent. They're not great at it because the hybrids are less viable, as we talked about, but they can do it to a certain extent, and this can cause the decline in allele frequency to become wider and narrower. Think of it as sort of the cline bleeding out or thinning out, flattening out, I guess I should say, as a result of alleles bleeding across it back and forth, as some of these hybrids do mate relatively successfully with homozygotes from either of the two populations on either side of the hybrid zone. All right. So, since hybrid heterozygotes have lower fitness than homozygotes on either side of the zone, allele A1 cannot increase in frequency in population A2, where allele A2 is fixed, and allele A2 cannot increase in frequency in population A1, or population 1 rather, where allele A1 is fixed. So that's what is called a tension zone. Okay, selection against these less fit hybrids minimizes gene flow between those two populations on either side of the hybrid zone. Now, the strength of selection against hybrids versus the amount of dispersal into the hybrid zone ultimately determines the steepness of the climb. If you have strong selection and low dispersal or low gene flow, you'll have a steep climb. If you have weak selection and high dispersal, high amounts of gene flow or migration, you'll have a fairly indistinct or flattened out climb. Okay, not as steep of a climb. All right, now let's talk about. Now let's talk about something called introgression. Okay, I need to go back to a blank slide here. Let's talk about something called introgression. Okay, so what if a selectively neutral locus, let's call it B, all right, is linked to allele A of our previous example? So here we have a neutral allele B that selection is not operating on, okay? And let's say that locus B has alleles B1 and B2, all right, B1 and B2. And let's say that these are fixed in populations 1 and 2 respectively. So, oops, sorry. Go back to my slideshow. All right. So let's say that these two alleles are fixed in their respective populations, okay? So again, locus B has alleles B1 and B2, and they're fixed in populations 1 and 2. So B1 is fixed in population 1, B2 is fixed in population 2. Now, if this neutral allele, or locus rather, B, is linked to the A locus, remember how we talked about genetic hitchhiking in a previous chapter, that sort of thing. If it's linked to the A locus, then they will have the tendency to hitchhike with the alleles A1 and A2, so that they will have a cline that's coincident with the cline of locus A from our previous example. All right. Now, if the B alleles are on a different chromosome that does not possess genes contributing to reduced hybrid fitness, they can actually move through the hybrid zone into the other population. So this diffusion of neutral alleles across a tension zone is called introgression. All right. So let's see how this might happen. So let's say that we have we have two parents. So we have an A1 parent, and again we're considering two genes here. So that parent's also B1. So it's homozygous for A, homozygous for B. Okay. And let's say it mates or crosses with an individual from the other side of the hybrid zone who is fixed for A2 and B2. All right. Okay. Well, what's going to happen here is that because one parent is fixed for A1 and B1 and the other is fixed for A2 and B2, is that all of their offspring will end up being heterozygotes. All right. So what you're going to get here then is all of the offspring being A1, A2, and B1, B2 heterozygotes. All right. Now, we're assuming that A1, A2 heterozygotes are bad. Remember, the B locus is neutral. It's not being selected for or against, so it can't be good or bad. Let's say the A1, A2 heterozygote, however, is suffering from hybrid inviability, okay? Heterozygote disadvantage, as it's called. Well, if that individual actually happens to mate, which can happen, okay, we're not saying they can't mate, we're just saying that they're mating at lower rates for the most part. If it happens to back cross with an individual, let's say A1, B1, B1, okay, it can back cross with that homozygote from one side of the hybrid zone. They can produce a wide variety of different genotypes in their offspring. But one of them that they can produce in particular is this, an A1 B1, B2 individual. All right. So this means that B2 is now in population 1, where it wasn't before, right? Up here, there's no B2 in individuals in that population. B1 was fixed, A1 was fixed, there's no B2 there. But because of the crossing here and the fact that you get heterozygotes in the second generation, and those can possibly back cross with individuals fixed for B1, you can have B2 actually ending up in this population now. And that's the main point right there, okay? That is called introgression. It could happen the other way around as well. You could have allele B2 moving into the B1 population, all right? Or B1 moving into the B2 population, I should say. So, because the neutral B2 allele has been disassociated from the deleterious or bad A1, A2 genotype through recombination, it can move across the tension zone and into the B1, B1 population. And like I said, allele B1 can also move into the B2, B2 population. So that is introgression, all right? It's sort of the bleeding of these neutral alleles across that hybrid zone by back crossing between hybrids and homozygotes on either side of it. Okay. All right. So last thing we're going to talk about in this chapter is the fate of hybrid zones. What happens to them? Well, a number of different things or scenarios can play out. First, hybrid zones can persist indefinitely due to selection that prevents elimination of clines. A hybrid zone could shift position to an area of low population density, for example, and because it's a low density area, there's a little potential for gene flow. Or you could have the environment changing and the environmental cline moving up a mountainside, down it, whatever, and that could actually cause the hybrid zone to move to the opposite edge of the population's geographic distribution, causing extinction of one of the populations. Sort of like a squeegee moving across a windshield, basically from one side to the other, if the analogy works. All right, second thing that can happen. Selection can favor alleles that reinforce prezygotic isolation among the two populations and eventually lead to complete reproductive isolation, where they're now fully two separate genetic species that are now simply incapable of reproducing with one another if individuals of both species do come into contact. Maybe now because of prezygotic barriers like mating preferences or whatever. Third thing that can happen to a hybrid zone. Mutation and selection may result in the accumulation of alleles that make hybrids more fit, actually, which may ultimately cause the postzygotic barriers to break down, right? So there are cases where it's been documented that hybrids are actually more fit than either of the two sort of parent populations that they came from. All right, in that case, those postzygotic barriers break down and you have the hybrid zone go away. And it's possible that the hybrids could spread left and right into their respective populations and wipe out the original two parent populations, okay? All right. Also, reproductive isolation of hybrids may cause them to evolve into a new species distinct from their parent species entirely. You could get a third species evolving where the hybrid zone was as a result of this sort of thing. All right, so that pretty much wraps up the chapter on species, chapter 17. And we'll move on with the next chapter, which I'm not sure what it is, but I'll figure that out here in a little bit, and that'll be it. All right.